Yeah, and if people are interested, actually, for our last minutes here, Nicola, we've gotten all of this data that he described onto Nemo, where we can really start to ask these questions quite directly. So this is a profile on Nemo, where each one of these boxes is a data set. And Nicola mentioned many data sets today, and each one of them is represented here. This first one is the 6-line bulk differentiation experiment with 3 controls and 3 ASD lines.
Right. Carlo, just let me just say that these are I didn't show this data, but these are related to the same project. And these are I showed you that the control and the SD lines were we only did the single cell RNA sequencing. I mean, I showed only the single cell RNA sequencing, but we did also bulk and differentiation. So I'm just and Carlo is showing the genes across these time course.
So the single cell data was from this time point here, day 8.
Exactly. Yeah.
And the 3 autistic lines and then the 3 control lines. And here we're looking at the expression of FOXG1, which of course Flora Vaccarino's group has seen misexpressed in autism since the 2015 Mariani paper. And they're connected it to the overexpression to ventral pallial fates. And so here we can also see almost exactly what she saw, that's almost a decade ago now, is that the lines derived from autistic individuals have higher FOXG1 expression through this time course. That's true in the differentiation experiment. And Nicola has also done a version of the passaging experiment with these same autistic and control lines. And there also, the autistic lines have greater FOXG1 expression. So right on Nemo, you can look across all of these data sets gene by gene. The other thing we can do, so here, this is the same collection of data sets, but instead of putting in a single gene, we've uploaded the lists of disease genes that Nicola spoke about. So hydrocephaly, lissencephaly, microcephaly, the different Safari scores. So for instance, here I have the list of Safari syndromic genes in ASD, and this is now the average, or in fact, it's the sum of expression of all the Safari syndromic genes across these same experiments. So they build over time in the differentiation experiment. So these syndromic genes are enriched in neuronal expressed genes. Something that's interesting, he mentioned, Nico mentioned the microcephalic genes. So if you look at the average of the microcephalic genes, it's the opposite. They're higher at earlier steps when there's still progenitor states around, and they go down in expression over time as cells differentiate into postmitotic neurons. So this is just to kind of give you an idea of what is possible at NEMO. And as much as we can use data, and specifically NEMO, if it works, to connect on findings. So we can not only talk about things, but we can see them in real time as we discuss possibilities here. Yeah. So I don't know if anybody has any questions, or in particular, or Nico, if you'd like to show something here.
I mean, I have a lot. It is very exciting, and yeah, I have a lot of comments, but maybe not to say.
Okay.
We don't want to. We don't want to keep people until tonight. But it's actually, but this is really interesting, because it is really interesting because now we are showing how the different risk genes which are associated with other disease, microcephaly or other disease, move in our control versus SD, right? So you can see how these risk genes may behave in another line, in the SD lines. So it's another way to connect, also to find features of different cortical disorders with connected with the ASD. Yeah.
So for instance,
it's also interesting, right?
Ask a question which to promote some thought on this. Yeah, for example, here, this image that we're looking at now. This is a mapping of microcephaly onto a data set, which we've talked about before, and so I
in the monkey data set.
If we mapped, right, exactly. If we remember before in this section, in this same setting, we discussed what would happen if you mapped cell cycle genes onto this, and we saw two different types. Yeah, exactly, two different types of cell cycle, and then everyone who's interested in cortical precursor cells, exactly. Yeah.
But Carlo, sorry, Ron.
When we well, let me just finish the thought then. So, when you say you can map risk onto this, shouldn't you be able to really predict certain features of phenotype now from your inferential models really quite precisely? It's not just a description, right? The description has reached a point where it has very tight predictive value. Right. For example, if to go back to Alex's work, the fact that there are two different ways of autism IPS cells behaving, I think is an absolutely critical result, right? Because it shows the spectrum of change, right? It's not just one change. One change could be associated with anything. Now you have, I don't know how to put it, but differential causality appearing.
Exactly. Yeah.
And I'm asking you in a way whether you can really take it down to proof, right? Not just a description. So this is different or that's different, but it starts here, and the molecular mechanism goes wrong at these places in the lineage, and this is what you see.
So
right. That's why when you look at the adult brain, different people have different numbers of neurons and different risk for different diseases. Right? It starts here, though. It's here you need to study, because it's here, if you know where it starts, you can do it very cheaply. Right? You don't have to grow these things for months and months and months. You can grow them in a short-term assay with large numbers of lines, which you seem to agree we need, although how many lines, I think, you know, if we really wanted to fund this work, we would want to answer that question quite precisely. But you see where I'm going? It seems to me that the answer to that, how many lines, is what are you trying to prove, right? And if you're trying to prove something mechanistic, then you have enough lines already.
Exactly, right. It is exactly the point.
Right.
That's why we use three. I mean, we use 3 lines, but we overcome this problem. You know, integrating the results of the obtained with only 3 SD.
Yeah.
The with the monkey data set. I mean, with the monkey can be also a human data set. I mean, we use the monkey data set because we profiled the pattern centers, and we were looking at difference in the pattern centers, right? It is to the best of my knowledge, is the data set in primates showing the molecular, where we profile the pattern centers. But then integrating this, how to say, comparing our results with a much more larger data set, like Jordan et al., now we could have a confirmation of what we were doing. So I totally agree. It's based on what we want to see. What you want to see probably tells you how many lines. I don't know if it is a real way to put the thoughts, but you, let's say that I think that you can overcome the problem of the number of lines. With these methods to integrate different or to look at different data sets. That's all. This is what I think is more can be more careful to say. I just want to say to Carlo that Carlo, here you are anyway pointing to this UMAP, but here is these are already late time point. You are missing all the early time point, which are all the late all the other DSLs are instead in this
yeah,
UMAP.
we have it both here. Yeah.
So yeah.
Yeah.
Because we talk about the involvement of neural stem cells in the cortical disorders, right? So the fact that
here's the
right, exactly, the fact that genes are involved
in particular.
the fact that genes are involved in the neurons, it's already known. I showed you that if you take all the risk genes associated with the neuropsychiatric disorders and you will check for enrichment of these genes in the macaque data, in the monkey data, you find that the majority of the genes are expressed in the neurons. We know that. The point is that,
right.
of course,
so we do syndromic from here, for instance, and then look. So this is ASD syndromic in Safari. Here you see at the end of these, but this is actually a great point here, is that it's highest at the ends of these trajectories where the most mature neurons are. But of course, there is some expression earlier back here.
Exactly. Right. That's the point.
So we need to be careful when we do these maps, right? Because mostly in the literature and in our thinking today, we map risk by the highest expression of that risk gene. Now, in the limit, that's very sensible. If the gene is not expressed in a cell at a time, it can't be the cause of the mechanistic problem. But it's not necessarily true that the highest expression of that gene is when and where the mechanistic problem is, right? So actually, Giorgio Quadrato's recent paper on SYNGAP1 kind of bring this up. It's an autism risk gene that's been studied for many years. It's part of the synaptic components and has been thought of and studied as a risk gene that confers risk for ASD by disturbing some of its function in the synapse. But her whole new paper is on how perturbations of SYNGAP1 actually mess with the proliferation of neural stem cells. So even here, when we say, oh, look, the syndromic genes are most highly expressed in the mature neurons, I'm not saying that, the syndromic genes aren't about perturbing the synapse, but we have to remember that many of these genes are expressed back here in the progenitors, and equally there could be causing problems even before the neurons are born.
Yeah.
And that's a harder thing to get a handle on because it requires us to get mechanistic insight, not just expression level. So it requires more experiments, of course, which everybody, especially like Alex's study, are really beginning to get into. And Alex, I'd love to work with you if you want to put in your father-son pairs along with your global map. That might be a fun thing to have in there. Then we could look at each individual pair of autistic father and son along all these different dimensions.
Yeah, that would be interesting also, yeah. And I want to say, like here, for example, also another hypothesis is that also Safari genes are quite complex in terms of like their impact, right? And syndromic, notably, you would expect them to see later in differentiation because also they are the one that will have the most drastic impact, right? So see if they are perturbed at the beginning of differentiation, they might have so much
you don't get a brain.
Would be impossible. My curiosity is like for more like genes with less effect on ASD, less association, right? Safari maybe score 1, 2, 3 might have like more subtle stage-specific effect that if they are completely mutated or not, they might have really a problem only at one stage during differentiations and maybe not have a drastic impact on brain, so not being that syndromic at all. But yeah, it's really interesting, and that's really like what we are trying to do also. Like we have a preprint online, like trying to build like regulatory network the same way like Nicola did also, is really the only way to understand to start understanding the hierarchy between genes, gene regulations, and how like tiny perturbation could sum up to large effects, all converging on ASD, for instance. And ASD, of course, we pick the most difficult disease to study, but to bring that together. And so having a multiple approaches to understand also the perturbation and everything, it's really interesting. Like also like for cell oracle predictions, that's also really interesting to see what could happen with some genes that are risk to disease, right? So that either on patterning or like on differentiation. That's really a lot of data to put together to understand really the full picture.
Right.
It's a lot of intersection to do. That's really interesting. Is a work.
We might want to have a session where we talk about these informatic tools as the main point of our discussion, I think. Because I think when you talk about Cell Oracle and the other tool you used here, I think they're becoming quite widely used. And just like when people first started doing UMaps, there was some kind of novelty to it, and you had to get used to being able to read these kinds of maps. the point that Alex is making now, I couldn't agree with more. I think that, you know, the proof is going to be in the network. And these network tools, if they are as advertised, they're really quite an advance. I'll encourage people to read a paper which has got Wolf Reich's name on it, where they use Cell Oracle to look at the mechanism of action of brachyury, which I found to be very helpful when I was thinking about this problem that Alex has just brought up, and that you raised well, Nicola, I thought, when you showed the informatic work. Anyway, cheers, guys. Thanks so much.
Okay.
Yeah, that's really important. So, like, one question I had maybe at some point, but we can discuss that, of course, later in another meeting. Like, first of all, this Cell Oracle, like, it's the perturbation seems to be a knockout system, right? It's kind of modelizing a knockout. And so, for example, I would have trouble relating it to my data when I see maybe dysregulation, so kind of a decreased expression, but not a complete knockout of the genes. So, for example, like, could Cell Oracle model also more subtle effect in terms of like just decreased expression, not full knockout of genes?
Sorry, would you say? Can you say repeat the question?
More a technical question on Cell Oracle, in the sense that here the modelization was cell knockout, gene knockout, right? So like
Right.
you
it does both. Yeah,
but it can also the expression, right? Slightly.
Yeah, overexpression. Yeah, absolutely. Yeah, absolutely.
That would be really cool to model to predict what I see, for example, in my data. Of course, you have cumulative effect of multiple genes that could be perturbed at the same time, right? But it's interesting to find. No, really, those are impressive tools and really hard to grasp and use correctly, right? There's so much they offer, but also so difficult to use and interpret. So it's really nice to see this study.
great, guys. Thanks so much for the talks and for the interaction.
Yeah, thank you for presenting.
Thank you so much. Thank you so much for this invitation. Thank you so much for this interaction. Thanks. See you next time.